crops won't die.
Let them die. I want that. I'm not feeding the crop.
Okay.
Saturday, Sunday, totally left. Them starving. Monday, I gave little food. And
View the
Tuesday,
chair, but chair अशी होते तेव्हा.
अरे यार, really यार. Saturday, Sunday, I didn't fed them. Monday, I gave feed for few hours. And now I am leaving. Today, I have not fed them. Tomorrow, I am not going to feed. Again, Thursday, I will see if my mood is good. Then
Why are you killing them?
by waste of things. If the bloody thing is there, then they will ask me to do something. Yesterday, sir came and I was doing analysis and you are maintaining the system. Yes, sir, I am maintaining. Are you checking the parameter? You are not checking, right? Why I will check? I won't check, na? Sir, no. Try to check at least once or twice in a week. Then you will get to know what is happening. See, these type of comments will come. I am not doing any analysis. I am not doing any study. But simply since I am maintaining the study, they are asking. He is asking me to do weekly once the parameter. Yeah, I don't do. I will say that I am doing.
Yeah.
Unnecessary things are all useless.
So in your next study, it is not needed.
That is not optimizing. You have to optimize that one, and it cannot be optimized. And that is the problem. Now it is working at its maximum efficiency, and which doesn't meet the minimum criteria of outside the
Mm.
full scale, whatever the full scale treatment systems.
So
Like
your
their
first
performance.
objective is over or you want to start?
Nothing is over. Zero. To start everything from the scratch. God knows. Chalo, bye bye.
Bye. Hello all. Welcome to week 10 interaction session for the course wastewater treatment and recycling. Let us wait another few minutes so that others would also join.
Okay.
Okay, let us start today's session. So I'll be sharing my screen now. So, welcome to week 10 interaction session for the course Wastewater Treatment and Recycling. So, I am Setu Lakshmi. I am a Prime Minister's Research Fellow doing PhD in Environmental Science and Engineering Department of IIT Bombay. So, today we will be discussing about some questions related to the week 10 lectures of NBTL course Wastewater Treatment and Recycling. So, I hope that all of you had watched the week 10 lectures before joining this session. If not, kindly watch all the lecture videos after this session and try to solve the assignment questions. This session will be helpful for you to solve the assignment problems. So, coming to a brief introduction, in week 10 you would have learned about the various conventional and advanced treatment methods of treatment of wastewater, and what is the series of operations, unit operations or processes which are followed in the conventional treatment. And also you would have learned about some of the advanced treatment procedures, and then in detailed you would have learned about constructed wetland for the treatment of wastewater. After that, there was also discussion about sequential batch reactors, sequential batch biofilm reactors, then membrane bioreactors, then moving bed bioreactors, etc. So there were discussion about all these types of wastewater treatment, and the advantages and disadvantages of each process. So our interaction session for today will be based on the MCQ questions on covering all these topics. So let us move on to the questions. So the first question is, which of the following is the major treatment process occurring in a wetland? So this is about constructed wetland, and the question is about which are the important processes which are happening in the constructed wetland. A. Adsorption, B. Bioaccumulation, C. Phytoremediation, D. All the above. Any guess? Anyone?
D, ma'am.
All the above.
All the above. Yes, it is correct. All the above. So, in constructed wetland, the mechanism of treatment of wastewater is due to all these parameters, that is adsorption, bioaccumulation, phytoremediation. Along with these three, there are other parameters also, which is this photoremediation, then biodegradation. So all these all other type or processes also help in the removal of polluted inside the constructed wetland. So this is about this question. Any doubt? Anyone?
No, ma'am.
Okay, so let us see the next question. Next question is the correct arrangement of units in a conventional aerobic sewage treatment plant is. So there are four options. So you have to pick the right sequence of operations. This is about the conventional aerobic sewage treatment plant. So
C, ma'am.
C, okay. Any other guess, anyone?
C.
C. Let us see the answer. C is the correct answer. Very good. So first there is screening process which removes all the floating and large debris present in the wastewater, followed by grid removal for the floating inorganic suspended solids present, then followed by primary sedimentation to remove the suspended solids mostly, then followed by the biological treatment. Conventionally, most of the areas or places we have the activated sludge process, which comprising of aeration tank and then a secondary sedimentation tank. So C is the correct sequence of operation, hence C is the correct answer. So any doubt?
No, ma'am.
Okay, then let us see the next question. The third question is, role of vegetation in wetland is A, to make it look attractive, B, to grow food crops for consumption, C, to remove contaminants through biosorption and bioaccumulation, and D, none of the above. So any answer?
C, ma'am.
C. Any
C,
other?
C.
Okay, let us see the answer. Yes, C is the correct answer. So first option is to make it look attractive. That is not an answer for this question because it is not scientifically correct. And then the next option is to grow food crops for consumption. Usually plants play a major role in removing pollutant from the constructed wetland, and usually such plants which are grown on constructed wetland for a pollutant removal is not used for the food as food crop for consumption, because there will be pollutant uptake by the plants. There are in some studies where some pollutant like heavy metals or industrial textile wastewater and all, there is uptake by the plant. So if we use those for the consumption, that will be hazardous for the animal which is consuming it. So it is not taken as a food crop for consumption. And the third option is to remove contaminants through biosorption and bioaccumulation, which is true, because plants have the capacity to remove pollutant with the mechanism of phytoremediation, which includes the process of biosorption and bioaccumulation. Hence C is the correct answer. Any doubt, anyone?
No, ma'am. No, ma'am.
Okay, so let us see the next question. So next question is, before that, I guess some of your friends are still trying to join, so let us include them also. Okay, let us continue. So the fourth question is, what is the correct order of phases in an SBR operation cycle? SBR is sequential batch reactor. So first is fill, react, decant, settle, idle. Then fill, react, settle, decant, idle. Then fill, decant, react, settle, idle, and then fill, decant, settle, react, idle. So 4 options are there. Any guess?
Option B.
B. B.
Option B, fill, react, settle, decant, idle. Okay, let us see the answer. Yes, option B is the correct answer. SBR is, as you have studied, in SBR our biological treatment happen in a single reactor where there are different stages, mainly 5 stages of operation. So there will be series of operations in which first we will fill the reactor with wastewater. There will be microbes or MLSs present in the system. Then after that there will be this mixing happening inside the system. In that time, that stage is known as react. First is fill. In the react stage, the microbes in the system will act on the wastewater and biochemical reactions happen, and the removal of contaminant occur during this stage. After that, all the content is given time to get settled. So that stage is settle stage. There will be separation happen where on top you will have the treated supernatant and below you will have the MLSs, the microbes or sludge. And after that, it is the decant stage, where you will, after keeping the sludge, you will decant the supernatant. That is the decant stage. Then after that a time is given for as idle time, where you will waste some amount of sludge in order to avoid the bulking inside the system. So some amount of sludge is gone as wasted sludge. So that is for that time is given as idle stage. Then again filling will happen, and then the process continues. So this is the ideal SBR. This is the ideal SBR process. So option B is the correct answer. Any doubt? Any doubt?
No, ma'am.
Okay, so let us see the next question. The components responsible for removal of contaminants in a wetland include A. Lining and vegetation, B. Vegetation and substrate media, C. Inlet and outlet arrangements, D. All of the above. Any guess?
B.
All of the above.
All of the
B.
above. B. Okay. Any other answer, anyone? Let us see the answer. B is the correct answer, because first option contains this term lining. Lining is not given for removal of contaminants. Lining is given inside the constructed wetland in order to avoid any any movement of
Leaching.
any leaching or any movement from outside to inside, any movement of wastewater or water from outside to inside or inside to outside. So in order to avoid the interaction from the surrounding, lining is provided. So this is not for the removal of contaminants. So A is not an option. B is vegetation and substrate media. Vegetation helps in the removal of contaminant by bioabsorption or phytoremediation or adsorption into the plant. The plants help in the removal of contaminants. Then substrate media helps in contaminant removal by this substrate media will act as a media for the growth of microbes, and the microbes present on the substrate media in the constructed wetland, they act on the wastewater when wastewater flows and biochemical reaction happens and the contaminant will get removed. That is one mechanism. Now another mechanism, the substrate media will adsorb the contaminant. Adsorption also will happen. So these mechanisms happen and substrate media will help in the removal of contaminants. Hence B is the correct answer. Then third option is inlet and outlet arrangements. They are not for the removal of contaminants. They are for giving inflow into the system and also taking out the outflow or the effluent. So option B is the correct answer. Shall we proceed to next question?
Yes.
Okay. So the next question is, in a typical SBR, aeration and mixing is avoided in A. First stage, B. Second stage, C. Third stage, and D. All of the above.
Third stage.
Third stage. Okay. Any other?
Third stage.
Okay. So let us see the next question. The answer. The answer is third stage. That is correct, because we have seen different stages will react, settle, decant, idle. Here it is asked aeration and mixing. So aeration and mixing is avoided in the third stage. Third stage is for settling. So mixing is avoided. Hence option C is the correct answer. Any doubt?
No.
Next is match the following question, where on the left side you have different processes, and on the right side you have the left side you have the mechanism, and the right side you have the process. So please match column 1 against.
C, ma'am.
C.
C.
Okay. Any other answer, anyone? Okay, let us see the answer. Answer is you are correct. Option C is the correct answer. So let us see the one by one. First is in option C, 1 is matched against B. So solid liquid separation, it is matched against B, that is membrane, which is true. In the membrane process, it is given for separating all the solid particles from the liquid content. So 1 is matched against B. Then 2 is against C. Oxygen supply. Oxygen supply is achieved through the process of aeration. Hence 2 is matched against C. And 3 is BOD reduction. So in the treatment process, BOD or biological oxygen demand, it is reduced with the help of microbes. You all know what is BOD, right? So BOD, it is a measurement of how much organic matter is present in the system. So when microbes act on the organic matter in the presence of air or without oxygen or with oxygen, I cannot use the word air in fact. So with or without oxygen, that is aerobic treatment or anaerobic treatment, this organic matter will get converted or oxidized to CO2 and water. So the third is BOD reduction against microorganism. Any doubt?
No, ma'am.
Okay. Then let us see the next question. So the next question is biomass growth in a sequencing batch biofilm reactor, that is SBBR. It is predominantly A. Fixed bed attached growth, B. Suspended growth in water, C. Attached growth on suspended carriers, D. All the above.
C, ma'am.
C. Attached growth on
D.
suspended carriers.
I don't know.
I'm sorry, can you repeat?
I thought it was D.
D.
Anyway,
Okay. So let us see the answer. Answer is C. Attached growth on suspended carriers. Here we are talking about sequencing batch biofilm reactors. So as you have learned in the NBTL lecture, in the SBBR there will be carriers present, and on the carriers this microbes will grow. So
okay.
So it is attached growth on suspended carriers. These carriers are suspended in the system, whereas microbes are attached to these carriers. And the mode of operation is this SBR mode, sequential batch reactor or batch mode. Hence the answer is option C. Is it clear?
Yes.
I just have one doubt, ma'am.
Yes.
Suppose we use fixed bed attached growth.
Yeah.
So the bed means biofilms can grow on the fixed bed.
Yes.
So why can't this also be this thing in the sequential batch biofilm reactor? Because the bed itself is a surface on which the biofilm film will grow.
Yeah, that is true. So here we will be talking in this context. We will be talking about this. For example, for that is this trickling filters. Do you know
Oh,
about trickling filters?
yes, ma'am. Yes.
Yeah, in the trickling filters, there will be one reactor where it will be packed with the carriers, and these carriers won't move. They will be fixed, and the microbes will get attached to these carriers. There will be no motion or no movement in the system. So those type of reactors are called as fixed bed reactors. So
Okay.
here we are talking about the sequential reactor where first we are like uh, here the advantage is that in the normal SBR the MLSs will be there. There is no media for them to get attached, and then you have the water. Then everything will be mixed, and then in the settle time these MLSs will get settled. So here in SBBR, the advantage is that there will be this improved uh, settleability of these MLSs, because as they are growing on this carriers, it the removal of these carriers from this wastewater stream is more easy, and
Okay.
it is known as SBBR because of its mode of operation. It is operated in this similar to the SBR reactors.
Thank you, ma'am. Thank you.
Okay. So let us see the next question.
One doubt, ma'am.
Yes.
Hello.
Yes.
Ma'am, focus on the suspended growth in water.
Yes.
It is aeration.
Sorry.
Uh,
What did you?
Suspended growth in water.
Yes.
It is aeration process.
Mostly it is aeration process. With the help of aeration, we provide the mixing in the system. Is it clear?
Okay.
Yeah. It can be operated in the anaerobic mode also. There are anaerobic SBR also, where there is no oxygen provided. In such cases, mechanical mixing is provided. Mechanical mixers are used.
In denitrification process.
In the case of denitrifying, like nitrification, denitrification process, yes. There are anaerobic SBRs also present. Is it clear?
Yes, ma'am.
Okay. So let us move on to the next question. So next is compared to conventional activated sludge process systems, membrane bioreactor operates at higher sludge concentration and lower reactor volumes. B, lower sludge concentration and lower reactor volumes. C, high sludge concentration and high reactor volumes. D, lower sludge concentration and high reactor volumes.
Aim.
A, high sludge concentration, low reactor
Yes.
volumes. Okay. Let us see the answer. Yes, A is the correct answer. Because when compared to the activated sludge process, which is a two-stage process, where there is one stage where aeration is provided and another stage where the settling operation is provided. In the membrane bioreactor, mostly I am talking about the submerged membrane bioreactor. In that we can operate it at high sludge concentration, because as we are using a membrane in order to separate the microbial biomass from the effluent stream, we can go for higher sludge concentration. And also, as it is a single stage process, the reactor volume is much less compared to the conventional ASP. Hence A is the correct answer. Any doubt, anyone?
No, ma'am.
Okay, so A is the correct answer. So let us see the next question. Next question is, which of the following is true regarding different configurations of MBR? So now we are talking about all the three configurations which you have studied. The submerged system of MBR, where the membrane is submerged inside the biological reactor, and then you have side stream system, where you have the biological reactor, and then the membrane is kept as a separate unit. That is known as side stream configuration. The options are, so the question is, which of the following statements are true? First statement is, low energy consumption is one of the advantage of side stream configuration. B, less rigorous cleaning procedures are required in submerged configuration. C, submerged configuration typically have high energy consumption. D, all of the above.
A.
A. Okay. Any other answer, anyone? Let us see the answer. Option B is the correct answer. Less rigorous cleaning procedure are required in submerged configuration. So usually the submerged configuration is better when compared to the side stream configuration in terms of biofouling as well as the energy consumption. So the difference advantages and disadvantages just of both the process you would have learned in the lecture. If not, please go through the lecture video. And it is mainly because of the, if you go into the mechanics or reason behind these two things, it is mainly because of the amount of pressure we need to apply for filtration to happen. The amount of pressure which is to be supplied is less in the submerged system than this side stream system. Hence the energy required for this system is less when compared to the side stream system. Also, as they are present inside the system, the biofouling would be less in the submerged system, as like inside the system there will be this aeration present, there will be the movement of microbes present inside the system, therefore the chances of biofouling are less, whereas in the separate system there are chances that the microbes will get attached to the system, as there is no way the no way the scaling can happen in this side stream system. Therefore, in terms of energy as well as in terms of cleaning procedure, we can say that the submerged system is better when compared to side stream system. Is it clear?
What is the advantage of a side stream configuration? Where is it used?
Usually, side stream system, I can say that when during the development stage, this was the first type which we configured. Then after that, the researchers have developed this submerged system. So nowadays, mostly we are going for this submerged system only because of the
Okay, so it's an
advantage.
older. Okay.
Yeah. So, any doubt, anyone?
No, ma'am.
Okay. So let us see the next question. So next question is the selection of choices of wastewater treatment system depends on
D.
A regulatory requirements, B installation and operation and maintenance cost, C intended reuse and disposal, D all the above. So I have got one answer as D. Any
D.
other? Yeah. Okay, D. So let us see the answer. Yes, D is the correct answer. So when we choose a wastewater treatment system, or when we design a system, we have to keep in mind about the regulatory requirements. What is our requirement? And also we have to be careful about the cost, because like uh, both the efficiency of the system and also the cost also matters as environmental engineers are concerned. So installation and operation maintenance cost is another aspect. The third is the intended reuse or disposal. So the when we choose the wastewater treatment system, we have to be careful about the reuse capability of the uh, of the units present, and also the disposal of the waste generated in the system. So for example, we can say that when we use the chemical coagulant, the all the sludge produced will be of the chemical nature, and the disposal of that sludge will be another headache. Therefore, we are going in more greener way nowadays by choosing more organic kind of coagulants instead of the chemical coagulant. So, such a way we have to be very much choosy about what material we use and what method we use. So, answer is all the above. Any doubt?
No, ma'am.
Okay. So, let us move on to next question. Next is about this moving bed biological reactor. The biofilm carriers in a moving bed biological reactor should be A. Capable of disinfecting microbes, B. Inert, C, of specific gravity significantly higher than water, D, having very smooth surface.
D.
Inert.
Inert. Okay. Any other answer? Let us see the answer.
C.
Yes,
B.
option B, inert, is the correct answer, because in the moving bed biological reactor also we have the microbes grown on carriers. So the carriers, the most important thing is that these carriers should be inert in nature. There should not be any interaction between these carriers and the wastewater. Therefore, option B is the correct answer. Any doubt? Shall we proceed to next question?
Yes, ma'am. Yes, ma'am.
So next is about next question is also about MBBR. So which of these would be better suited as carriers that provide surface for biofilm growth in an MBBR? A, high density polyethylene, which is HDPE of density 0.95 gram per cc, then melamine polymer of density 2 gram per cc, then polypropylene of density 0.87 gram per cc, silica gel of density 2.6 gram per cc.
A. A
A.
answer.
A.
Okay. So those who have said A, could you please tell me why you have chosen A?
Because density of the media is lower than water.
Okay. But then why haven't you chose this polypropylene?
It is
Polypropylene.
polypropylene is density is very low, so
Okay.
that it is carry forward to the through the MBBR bed.
So the requirement is that the density of the carrier should be nearly equal to water. Is that correct?
Water. Yeah.
Yeah, that is correct.
Okay.
So it should be nearly 1 gram per cc. That's why we go for high density polyethylene. Is it clear?
Yes, ma'am.
Okay. So then we shall proceed to the next question. Next is a match the following type. On column 1 you have different treatment processes, and then column 2 you have mm, the sequence. So you have to match column 1 against column 2. Any answer, anyone?
A answer. A answer.
A. Okay.
Yeah, I think it's A.
Okay.
A is right.
A is the answer. Okay. So let us see. Yes, A is the correct answer. You are right. SBR means it is a sequential batch reactor. It is matched against C. So that means that SBR, when we explain, it means that the
It is batch reactor.
yeah, it is the configuration is same as that of an activated sludge processes, but the processes are operated in batches in the same reactor. In ASP we have two different reactors, but in SBR it is done in same reactor. Then you have 2 against D, that is SBBR against D. That means that it is same as that of an SBR, but the biofilm is grown on a carrier media. Therefore 2 against D. Then the third is MBR. It is against A, which means that in the membrane biological reactor or membrane bioreactor, we have an ASP, an activated sludge process. Also we have a membrane filtration equipment which is kept inside the, instead of a settling unit we have the membrane filtration equipment in the aeration tank. So MBR is matched against A, and then you have MBBR fourth one, which is against B. That is, it is a MBBR means it's a conventional biological treatment where the microbes are doing the pollutant removal, and the process is that it is an attached growth system where the microbes are attached to the carrier media, which we have already explained in the previous question. So hence, the answer is A. Is it clear?
Yes, ma'am.
Okay. So next, I guess this is the last question. You don't have any numerical question for today's class. So this is the last question is, the degree of fouling in an MBR depends on A, feed and biomass properties, B, membrane properties, C, operating conditions, and D, all the above.
D, ma'am.
Operation
All
condition.
the above.
Only operation conditions. Okay. Any other answer?
D, ma'am.
D. Okay. Let us
All of the above.
Let us see the answer. All the above is the answer. The degree of fouling in a membrane bioreactor, it depends on the type of feed, then the biomass properties, then also the membrane properties also affect the fouling. The material we choose for membrane, then the other parameters, operating conditions, the pressure, flow direction. So everything occurs, or everything affects these parameters affect the degree of fouling in an MBR. Hence, all of the above is the answer. So this is all about. This is the last question. So with that, we end our today's class. Any doubt, anyone?
We were to discuss
No,
something this week.
Yeah, we were. I still remember that. So.
Okay.
So I don't think that all those students, or all of our friends who were present last time are present today. Only
Okay.
we both are there, I guess.
I guess so.
Yeah.
I just did a bit of the thing. I mean, if others are interested, we can just.
Sure.
Can I just tell them, ma'am, if you don't mind? If you
I
got time.
Yeah, I don't mind. In fact, I would love to listen to you, your opinion and all. Like in that process, we all can learn together. So,
Actually,
can you please explain or start from our question or statement, and then
okay.
yeah.
Actually, the last time a doubt was raised regarding membrane disinfecting and disinfecting the water and then passing it on for the membrane, what you call membrane separation.
Yes.
So that was the basic idea.
Yeah.
Uh, that because we say disinfection first and then membrane,
Yes.
but quite a few in the industry do the membrane separation first and then disinfect later.
Yes.
So what I have found out is they do disinfect earlier to prevent membrane fouling. But if chlorine is used, then you have to remove the chlorine if at all certain types of membranes are used, especially cellulose acetate.
Okay.
So what they do is when you pass chlorine or ozone, in the case of ozone and chlorine, these are both oxidants. So you have to remove chlorine by dosing it with SMBS. And the case of ozone, like in the pharma industry, we remove it by using what we call UV light. We destroy the ozone by using UV. So that's neutralized basically that way. But that is the basic the thing in this. And I think that the book you had suggested, Metcalf,
Yeah.
Metcalf, yeah,
Makeup.
they were suggesting disinfection prior to membrane separation. So uh, that also takes place. One small doubt was, man,
Yeah.
I mean, we are now in the 11th week. The thing
Yes.
I do not find any because most people are from the industry would have come across the sand filter,
Okay.
and after the secondary stage, after the secondary stage, usually what we do is we go into a sand filter
Yeah.
and then disinfect the water. There is no mention anywhere about the sand filter.
Okay.
Why is that? Is that any reason that it has now been, it's an old technology and no one wants it? Or
Actually, I can tell you that as you know that the sand filtration is not effective for the emerging contaminant removal. It like the it cannot remove the suspended sorry the dissolved ions or dissolved metals and other like other pollutants like the other emerging contaminants like pharmaceuticals and all. So nowadays we are more concerned about the emerging contaminants, right?
okay,
So the sand filtration has the limitation because of its pore size. So
fine.
when we consider other processes like ultrafiltration or this reverse osmosis, all those are more effective when compared to sand filtration in removal of all other macro as well as micro molecules.
Okay.
So I guess that is why nowadays we are not going for sand filtration, as in today's world we are talking about water recycling more than the water discharge into the water bodies after treatment.
My the thing is, man,
Yeah.
because when you're using high quality membranes like RO,
Yes.
UF, and nanofilters and so
Yeah.
on, basically any filtration, what we try to do is to reduce large size particles moving down to the finer filters.
Yes.
So would not sand filter help in reducing the large particles?
Sand filtration helps, but then like it's like then you are suggesting that after sand filtration shall we move on to the other processes?
Yes, that's
Yeah,
what I'm
that
suggesting.
can be done, but then we are increasing the stage of operation, right? Like
But you're saving a lot on cost.
that is also there, but
You'll
then
be saving a lot on cost.
yeah, I understand that. So that is why like when you do the cost analysis altogether, uh, like for that we have to do this cost analysis and all, like which is more effective, so which is more economical and all. So and also of course we are not interested in multiple stage of processes.
Okay.
Maybe all these are reasons why we are nowadays we are not talking about sand filtration.
Okay.
Yeah.
Thank you, ma'am. Thank you.
Yeah, thank you so much for your interesting contribution.
Thank you, ma'am.
Yeah.
Thank you.
So that's even I also have your same opinion when we are talking about this using chlorination before membrane processes. I also have read about it, not much in detail because of my time restrictions and all. But somewhere I have read that if you are using chlorination before membrane process, you need to remove the chlorine by methods, for example by carbon adsorption and all, before
Yeah.
supplying it to the membrane bioreactor.
Carbon absorption is there, ma'am, but the faster method is introducing SMBS. Uh,
SM.
yeah, sodium metabisulfate.
Okay.
That's
Yeah.
usually faster.
Ah.
But again, you're introducing a contaminant which you have to remove later on.
Okay.
So carbon absorption is a better alternative for than SMBS, I would say.
It is very cheap and carbon absorption is very costly and continue it needs to the continuous changes.
Yeah,
Okay.
it's true.
Sure, that might be true. So, so anyway, I also have your same opinion. Though I have not read much in detail about that because of my time limitations. So it's good that you have put such a great effort in this.
No,
Thank
no,
you
no,
so
no.
much.
Thank you. Thank you.
Yeah. So that's all about today's session. Now we have another two more classes, I guess, for week 11 and week 12. So
Okay.
let us all meet on next Tuesday then.
Yes, ma'am.
Shall
Thank you.
we close today's session?
Yes, ma'am. Thank you.
And happy Dasra to all of you.
Happy Dasara to ma'am.
Happy Dasara to ma'am.